Bismillahirrahmanirrahim, and welcome back to this next video. And this is the 11th video in the series of videos on immunology. In my previous videos, I've told you that there were two schools of thoughts. One, they was believing in humoral immunity, and they believed that there are soluble components in the body that provide immunity against the pathogens. The other school of thought, they was believing in the cell-mediated immunity, and they believed that there are cells in the body that provide protection against the pathogens. And in my previous video, I've told you that at the end of the day, it was believed that we need both the humoral immunity and the cell-mediated immunity for a complete immune system. Now, it was established that we need the humoral as well as the cell-mediated immunity, but the early immunologists, they were, you can say, there was an enigma going on that what determines the specificity of the immune system against an antigen. And when we use this term antigen, it can be any substance that elicits an immune response in living organism. So they were confused that what is going to determine the specificity of the immune system against the antigen. The second thing that the early immunologists believed that the immune system only gets activated when the infectious agent enters into the living organisms. But around 1900, Jules Bordet at the Pasteur Institute, he extended the concept of immunity beyond the infectious diseases. And he said that non-pathogenic substances like the red blood cells from the other species, they can also activate the immune system. What that means is that the non-pathogenic substances, just like the infectious diseases, excuse me, the non-pathogenic substances like the RBCs, just like the infectious diseases, they can activate the immune system. Now, what they believed, the study of the Jules Bordet and others, they believe that the foreign materials, whether they are infectious or they are non-infectious, they have the ability to elicit the immune response. That means that they can be responsible for the production of the antibodies and the other components of the immune system. Now, Landsteiner, he believed that almost any non-self organic chemical, this is important to keep in mind that we are talking about the non-self things. That means that the chemicals that are not present in our body do not recognize them as their own. So, Landsteiner, he believed that almost any non-self organic chemical has the ability to induce the production of antibody against those specific chemicals. So if a pathogenic organism is entering into the body, the antibodies will be made against the pathogen. If a non-pathogenic substance is entering into your body, the antibodies that will be made against those particular non-pathogenic substances. Or if they say, for example, if the RBCs, they are entering into the body, the antibodies, they are going to be made against these RBCs. Now, they believed, like these Landsteiner and the other studies, they let us believe that these antibodies, they have got an unlimited range of reactivity, and they can even be synthesized against the compounds that has recently been synthesized in the lab. So they said that the antibodies, they have got an unlimited range of reactivity, and they can be made against theoretically any kind of the compound, whether that is infectious, that is non-infectious, whatever the case is. But if that chemical is non-self, the antibodies, they would be made against these chemicals. Another important thing was that this is going to tell us about the range of the antibodies. Another thing they believed, or they proved, that these antibodies, they can actually differentiate between the smallest details of the molecules. So if there are two proteins, for example, two foreign proteins, and they only differ by a single amino acid, the body is going to produce two types of the antibodies against these two different proteins. What that means is that these antibodies, they are very specific in nature, and they can even note the smallest details that is differentiating the two substances, proteins for example. Now, to account for this particular specificity, to account for this unlimited range of reactivity, two theories they were proposed. The first one that was known as the selective theory, and the other one that is known as the instructional theory. And in the few videos to come, I'll be focusing on these selective theory and instructional theory in detail, and I tell you that what the selective theory means and what the instructional theory means, and how these two theories, they help us led to our current knowledge in the field of immunology, when we talk about the specificity and when we talk about the range of reactivity of antibodies. So if you like the video, please subscribe to my channel, hit the notification bell, and do share it with your friends. I'll see you in the next video.